So, boys and girls, we stopped last time at the structure of chromosome, correct? See, before, as I last time also told you, that when you are born, God has gifted you the reproductive organs. Boys have penis, testes, girls have ovaries. But is it that at the age of first or at the age of second, boys and girls can start reproducing? Do the reproduce at that age? No. They have to go under the adolescence first. Correct? Where the secondary characteristics start showing up. And then one has to attain the adulthood. That is at the age of 19. We see that it's an adulthood. Where now your reproductive organs are matured. And then they are capable of, or he or she is capable of reproducing. Similarly, in the cellulose, once a cell is born, it cannot undergo the division just like that. Just like that, it cannot go the division. It has to mature first. It has to mature first. And once it is mature, then one is ready. Now, what is this phase of getting mature of a cell is known as? This is known as interphase. Okay. Actually, the entire cell division is based on two things. It is called as cell cycle. Okay. Boys, can you see this? What I've written?
Yes,
Are
sir.
you all huh?
Yes, sir.
Girls?
Yes,
Can
sir.
everybody see
Yes,
what
sir.
I've
Yes,
written?
sir.
So the cell cycle is made of two things. One is interphase and other is cell division. Okay. Now, in our chapter, we are going to study two types of cell divisions. Okay, we're going to study the two types of cell divisions. Those two types of cell divisions are mitosis and meiosis. Right? Mitosis is simply meaning breaking into two parts from one. It is breaking of a thread into two parts. Okay? Like we have seen in the binary fission. And from one cell, two cells are produced. Binary fission is a very primitive mitosis only, very early stage of mitosis. Right? And that is in the prokaryotes. Whereas in eukaryotes also show binary fusion. But the higher eukaryotes, that is, who have body structure, okay, they do
No, your
not show.
voice is breaking.
My voice is breaking. Is it,
No, sir, it
is
is
it
still
clear?
breaking.
See, guys, you have to get your interconnections proper. Okay. Now, mitosis, before we go here, mitosis, mitosis and meiosis we are going to see. Just tell me the spelling of meiosis. What we can do is that book and share together. Starting much, you're very mass, just keep a pack in between. But you don't, and keep your, you should take your both this side and keep your text both. Okay. Now mitosis is shown by somatic cells. Somatic cells. What they are also called as autosomes. They are called as autosomes. Okay. Whereas meiosis is shown by gonads. It's a term used for testes and ovaries. Okay, it is a general term, and autosomes and the somatic cells, they are found all over the body. Both external and internal organs. Okay. Now there are certain things which I want you to know from your textbook, which are many a times asked in the exam. Yes. Now, this mitosis, this much you should know first. Okay, and meiosis, that is carried out in the testes and ovaries, is only for production of gametes, that is sperms and ova. Whereas here in mitosis is for repairs, growth, and replacement. Okay. Now, cell division is a continuous process. It's a continuous process because that is mitosis. Because our cells get older, they die, they have to be replaced. When the cells are born, the lateral and the vertical growth takes place. That is, you were just a baby, now you're a tall adult, correct? That's a change that has been brought because of the cell division which has taken place, and it is still going to go on. Many times you may have a trauma, some injury. I'm sure you girls might be cutting the vegetables or chiseling something, you cut your hand, while making rotis, you burn your hands. What happens to those cells? After some day, you find the same new skin. The reason is that it is replaced. But then you will ask me, sir, do all the organs have such a facility of continuous cell division? Then I would say no. There are certain exceptions in the law. There are certain exceptions in the law. That is your brain cells, that is nerve cells. Nerve cells. They never divide. They never divide. Absolutely never divide. Your liver cells, once in a year, maybe they may divide only once every 1 or 2 years to replace the damaged cells. Liver साल 2 साल में एक बार ही करता है, once or once in 2 years. This undergoes. Please note why I'm giving you is that many times short answer में questions आता है, filling the blanks के लिए, nice of orange के लिए, half mark के लिए. Okay? या नेम द फॉलोइंग के लिए जो 10 नेम द फॉलोइंग आता है ना, 5 मार्क के लिए हाफ मार्क का एक नेम द फॉलोइंग, उसमें ये बहुत बार पूछा जाता है. Your surface skin are in continuous loss. See, when you are in winter, you scratch, you get that whitish powder. What is that? Your squamous epithelial cells are being removed. Your squamous epithelial cells are being removed. Okay, so they have to be replaced. Okay, they have to be replaced. Sometimes due to the overheat, the sunburns are there. So that time you need to be replacing them. So your skin is the, you can say that your skin has got a meristematic tissues like that of your plant. And they have only one textbook. And what I've done is that they can just keep it. The overtime she cannot come back. So just for the sake of another 10-15 minutes. After the explanation, you can go back. Okay. So, skin tissues are in squamous epithelium, the organs of the continuation. In plants, the meristematic tissues at the apical, I hope you know the apical, lateral. You know those meristems names, or you want me to give them? See, in plants,
No, sir, we know.
Meristem. These meristems are apical, lateral, intercalary, okay, and cambium. So at these places, apical is the tip of the root and the shoot. The lateral is in between the nodes and internodes. I hope you know what is a node and internode. Say on a stem, at regular intervals, you find the outgrowths. Especially if you see the hibiscus plant or the rose plant, you will find these are called as nodes. And this is called as internode. So at these places you will find them in the center of the stem. You find that such concentric circles being created. If any carpentry work is going in your house, I'm sure you must have seen such cuts, circles on the wooden plank or on the wooden chip, right? So these are the places where the continuous cell division is taking place, right? Now we come to the part of this chapter. Chapter's most important part is the cell cycle. Do you want me to run? Meet fellows?
Yes,
Do you
sir.
want-
Yes, sir.
Or have you noted down? Boys and
Sir,
girls,
done,
have
sir,
you
wrote
noted down? Am I audible to you?
Yes, sir.
When I asked you, have you noted down these things?
Yes,
Yes,
sir.
sir.
Yes.
Okay. See, before a cell has to do the cell division, before a cell undergoes a cell division, before a cell undergoes, it starts preparing its. Say, if I want to tell you that there is a mother who has got two twin daughters, a couple is there, who has got twin daughters. So they would not do any partiality between them. Say they would not do that. If a chocolate is brought, only one is given to one girl and other is being starved off. It is not that. So when as they grow up, for the security of their future, the mother starts preparing everything in. Correct, no? इसको अगर गहने लिए तो इसको भी गहने लिए, इसको साड़ी लानी तो इसको भी साड़ी लानी. Right? And then they start preparing for them, and the D-Day comes and they get married, and they give the share, and they see that the daughters start their new life. So mother cell is there. The mother cell, before getting into the cell division, starts preparing itself for the cell division. And this is done under the cell cycle. In this cell cycle, you have these two different phases. One is an interface, and other is a cell division. This cell division will consist of mitosis. We are talking about the. So this mitosis will consist of following things: prophase, metaphase, anaphase, telophase, and cytokinesis. Together, all these are known as karyokinesis. Together, they are known as karyokinesis. Okay. Now we come to the interphase first. This phase is called as resting phase. This phase is called as resting phase. Now, why it is called as resting phase? Because during this phase, you literally cannot see. Anything happening inside the cell. Under the microscope, you will find nothing happening inside the cell. But in reality, a lot of physiological activities are going on. And these physiological activities are divided into 3 checkpoints. You know, like before a rocket launch, they check all the 3 parts of the rocket. Then they say check 1 phase is finished, check 2 phase is finished, check 3 phase is finished, and the countdown starts. And at 0, the rocket is launched. Similarly, in the interface, you have 3 types of phases going on. This phase is known as G1 phase or gap 1 phase. Then this phase, second phase, after the gap 1 phase, you have S phase. And then you have gap 2 phase. And then afterwards, after the entire check is done, the mitosis will start, which will have prophase, metaphase, anaphase, telophase, and cytokinesis. Now, what will happen in this G1 phase? In this G1 phase, everything that is inside the cell will start to duplicate itself. अब उसको duplicate क्यों करना है? तो जो नए आने वाला जो सेल है उसको इक्वल क्वांटिटी में सब कुछ दो, बराबर। तो इन दिस phase what will happen, cytoplasm increases in quantity, increases in quantity. Then chloroplast in plant cell duplicate. Mitochondria and PR duplicate. That means all, just remember, all cell organelles will duplicate. Okay? All cell organelles will duplicate. And most important thing will happen. DNA and RNA will be synthesized. Now, why do we require RNA and DNA? RNA is required to prepare the proteins. And the proteins are and those proteins are needed by DNA to prepare the genes. So we need DNA also and we need RNA, and RNA will only prepare DNA. RNA से ही DNA बनता है. Okay. So you should know that this DNA and RNA are very, very important for us. Okay. Then the S phase. Now check one point is over. Cell has prepared and the cell gives the green signal. My G1 phase is over, check one over. Proceed to check two. तो S phase में क्या होता है? S phase में क्रोमोसोम replicate यानी अपने आप को ग्रो बनाते हैं. Sister chromatics. It converted to sister. इसको replicate भी बोल सकते हो या duplicate भी बोल सकते हो. Why it is going to get duplicate? I will tell you. ये स्टार मार्क करके बहुत बोल्ड में लिखूं. In mitosis, number of chromosomes are maintained same throughout the cell division. Yes? Boys and girls, can you see what I've written? Can you see what I've written out there?
Yes, sir. Yes.
What does it mean?
Yes, sir.
What does it mean? See, we have mother and we have father. Okay? Now, the bachchu. You know that we have 46 in humans, there are 46 number of chromosomes. Either 46 number you can remember or 23 pairs. Okay. So even the offspring, now 13, when the offspring is born, he gets 13 from mother, number of pair of chromosomes, and 13 from, okay, not that much, 23 and a half. 12 and a half, 12 and a half. Wait, 23 and a half कितना होगा? He gets half the chromosome number from mother, or he gets half the chromosome number. Okay, or we can say that he gets 10 number of chromosome from mother, and he gets 10 number of chromosome from father, and he gets 2n number of chromosome. This 2n number always will represent 46 chromosomes. ध्यान में वो जो मैं लिख रहा हूं, please note it down. This is INP. You should know that what is meaning of 2n. 2n is 46 number of chromosomes or 23 pair of chromosomes. Is that understood? Meaning is same. So in mitosis, why does a chromosome in the S phase have to get duplicated? Because in every step, except in prophase, if there were 46 chromosomes, if my cell of skin is dividing and it is going under the mitosis, and my cell has got 46 chromosomes, then while doing the division, its prophase will also have 46. When it enter into metaphase, that time also the number of chromosomes will be 46. When it will enter into anaphase, that time also it will be 46. When it will enter into telophase, it will be 46, and ultimately in cytokinesis, it will be 46 chromosomes distributed to, or 23 pairs of chromosomes distributed, which will then again form the chromatin fibers. Okay. So in this S phase, the chromosomes duplicate and more amount of DNA and protein is synthesized. More DNA is synthesized. More DNA is synthesized. Okay? Why? Because now we want to create that DNA into where? Onto this chromosomes. The chromosomes are going to require it. Those DNAs to maintain that hereditary characteristic and get converted into chromosomes. Once this phase is done, we enter into G2 phase. And in this G2 phase, again, lot of protein that is RNA and DNA is synthesized. Okay? Is synthesized. And now, once this all is done, once the interphase is totally completed, it has finished the G1 phase, S phase, and G2 phase. Now our cell is ready to undergo karyokinesis. What is karyokinesis together? That is starting from prophase, metaphase, anaphase, telophase, and cytokinesis. Meet fellows, Bachas, have you understood up to this much?
Yes,
sir.
sir.
Everybody has understood?
Yes, sir.
Yes, sir. Yes, sir.
Girls, I did
Yes,
not
sir.
hear your chorus.
Yes, sir.
Yes.
Yes, sir, understood.
Now, the cell is going to enter into metaphase. Mitosis. Why this? Actually, there is no such thing such as early prophase, latter prophase, early anaphase, latter anaphase. No such things are there. It's only prophase, metaphase, anaphase. Okay. But for your understanding better, our book has divided the prophase into two parts. Now pay attention to my diagrams and pay attention to what I am telling you. Okay? Okay, first I'll explain you the phase and then I'll tell you the answer. Can I rub all these things which I have written on the board?
Yes, sir.
Yes, sir. Sure, sir.
Okay, girls, do I remove it? Girls, can you please tell me, do I remove this?
yes,
अबे
sir.
भाई लोग मेरे बोलो, I'm talking to walls. ये मैं निकाल दो या रख दूं?
Done.
किसी को अगर लिखना है तो लिख लो और फिर मेरे को बोल दो कि सर निकालूंगा ये बोर्ड में से. I have to start teaching the prophase and metaphase today at least at any cost. I want to complete it. So I take the consent that you have given me the consent to remove it. Now, I will be explaining you both plant cell and the animal cell simultaneously. Yeah? See, I told you in the beginning, while teaching you the structure of chromosome, that as soon as the centrosome with the centriole will divide, it triggers the cell division. It means it is a start of the cell division. So what the cell will do is that it will divide the centrosome with centriole into two. Okay. Once this start happens, so what is the first point of the prophase? We are doing prophase. Prophase, early prophase. Okay. First point will be centrosome with centriole divides into two, now called as aster. Asters. Okay. This is your first point of the early prophase. The chromosomes become distinct. Chromosomes become distinct and appear. Okay? And now third, asters will move to the poles. Aster will move to poles. Okay? In plant cell, there is no centrosome. So, what will happen? Only the chromosomes will become distinct. Correct?
Yes, sir.
Then now we come to the latter prophase. Latter prophase. Here, what is going to take place here? These are the asters. Correct? Now the nucleus will disappear. Now the nucleus will disappear. The nuclear material will get disappear, and the microtubules will start appearing. And these are called as spindles or spindle fibers. To these, the chromatids, here chromosome become distinct and appear and break into sister chromatids joined by centromere. I forgot to write that point. क्रोमोसोम डिस्ट्रिक्ट हो जाएंगे, वो अपीयर हो जाएंगे किसमें? अर्ली प्रोफेज में. वो ब्रेकडाउन किसमें हो जाएंगे? टू सिस्टर क्रोमैटिड्स में. और वो टू सिस्टर क्रोमैटिड्स कैसे ज्वाइन होंगे एक दूसरे को? विद द हेल्प ऑफ सेंट्रोमियर. वीट के बच्चों समझा क्या? अर्ली
हाँ सर,
प्रोफेज
समझा सर.
मैंने बताया है, उसमें ऐसा है, chromosomes become distinct, they appear, chromosomes break into sister chromatids. वो sister chromatids जो मैंने सिखाया तुमको during the structure of chromosome, उसमें वो convert हो जाते हैं और sister chromatids एक दूसरे के साथ कैसे जुड़ जाते हैं? with centromere. फिर latter prophase में क्या होता है? in animal cell, the nucleus disappears. The nucleus disappears. The nuclear membrane breaks off, and instead of that, what happens? The microtubules, called as spindle fibers, they are produced, and then something very important happens. The sister chromatids join to the microtubule with centromere at the center. Okay. They join to the spindle fibers. The sister chromatids join to the spindle fibers. How? The centromere joins to the centromere joins to the spindle fibers. The centromere joins the chromatophore joins to the, and here in the same thing happens. There is a spindle fiber formation. And like here in the insect, in the animal cell, the sister chromatids they join like this. In the plant cell, okay, it is beautifully, don't worry. Just see and then afterwards draw this to the help of your textbook, which I'm going to do. Have you understood this much in the meat?
Yes, sir.
Yes, sir.
Girls, have you understood? Girls, have you
Yes, sir.
Okay. So write down the points now. I don't know the place. I'm dictating the points of the lateral prophase. So please write down the points of the lateral prophase. The nuclear material disappear. The nuclear material disappear. Nuclear membrane breakdown. Nuclear membrane breakdown. Microtubules called as spindle fibers appear. Microtubules called as spindle fibers appear. I'll repeat all these 3 points again. Check out whether you have written it or not. The nuclear material disappears. Nuclear membrane breaks down. Microtubules called as spindle fibers appear. The sister chromatids join to the spindle fiber, which centromere at center. Has everybody understood this prophase?
Sir, join to it.
Has everybody understood the prophase? Now, see,
Excuse me, sir.
Yes, beta.
Sir, can you repeat the 4th point?
Okay. The sister chromatids join the spindle fiber with centromere at the center.
Yes, sir. Thank you, sir.
Have you understood this much? Written this much?
Yes, sir. Thank you, sir.
Now, boys and girls, in board, they'll give you either the picture of early prophase. Okay. How the questions are going to come, you should know. They'll give you a picture of early prophase. They'll ask you to identify. They'll ask you to name the asters, spindle fibers, nucleus, all those things. And then suddenly they will ask you, what is, draw the step. They will ask 2 questions. Draw the step prior to this and proceeding to this. Now what generally you people do is that you write interface barabar. Before this is interface. So you draw that interface wala diagram. And the next phase what you draw is of the lateral profiles, which is wrong. And there you lose your mark. As I told you, for your understanding, the book has created the early profiles and lateral profiles. In reality, there is no lateral profiles and early profiles. Directly profiles and then metaphors. So in such cases, what you have to do? In such cases, you have to write that the proceeding phase is the metaphase, draw it and describe. So such questions. Always remember these tricky questions may come for you from between the prophase and anaphase, where there are early and latter phases. समझ में आया आज का टॉपिक?
हाँ सर.
And the last part I want to tell you, which I want to finish off today, is the metaphase. Let me first draw the diagram and then show it to you. Now in metaphase, now I'm just drawing this aspect with the star marker for your understanding. Okay. The same thing you have to draw for the plant also. Now, this is called as equatorial plane. This is called as equatorial plane. So the system chromatids, they join along the equatorial plane in the so in straight line, this is only the metaphors. All that you have to write is over here, draw this diagram and write that. The system chromatids align themselves or line up at the equatorial plane. Now, these are all your. This is where we are going to stop today. Right? So, I thank you very much for being with me. Have you all understood? Now I'm talking to my NEET fellows.
Yes sir.
Yes, sir.
Right, boys and girls? Any difficulty in understanding these things?
No
Cell
difficulty,
cycle?
sir.
No sir, no.
No, sir.
Girls, any difficulty, any doubt in cell cycle?
No, sir.
No? Okay. Then any difficulty in understanding early prophase, latter prophase, and metaphase? Yes, Parth and the same other fellow who are very interactive, please tell me, have you understood these pages?
Yes sir.
Yes,
Understood.
sir. Yes, sir.
Girls, have you understood these pages?
Yes, sir.
Girls? Okay. Thank you very much for being with me. Right? Those who
Thank
have
you.
the textbooks, wait, listen, there is a homework. Those who have the textbooks, you can take the screenshots if you want to. You should. Push screenshots later also. You have to draw a beautiful diagram of cell cycle and explain it in your own words. Question number 1, draw the diagram of cell cycle and explain it in your own words. Second, draw the diagrams of interphase, early prophase, latter prophase, and metaphase with proper labeling and explain each phase in your own words. Use of rounder while drawing the diagram is must. Do not oblige me by just drawing a diagram. Please draw it very nicely, very clear, and label them very nicely. I am emphasizing you, my boys and girls, all the diagrams of each chapter. You should keep, if you have got those last year's notebooks, prepare one small 200 and 300 pages notebook. But each chapter wise, you should practice all the diagrams at least 5 times to make your labels pakka, because I'm telling you, your 2 to 3 marks in your section 2 are eaten up by your labels. Either they ask you labeling for 1.5 mark, that is half mark, or they ask 4 labelings, half mark each for 2 marks, or they ask 6 labelings for 3 marks, or 2.5 labelings for 5 marks. 5 labelings for 2.5 marks. Please, I have seen people losing their entire thing. Rest all theory out of 7 out of 7. Labeling में गया पाना. So please don't do this. I would like to see your homework when sir puts up our next lecture.